On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
In this episode of the Life Science Success Podcast, we'll be discussing liquid biopsies for early detection of cancers. I'm joined today by Dr. Sam Hanash from the MD Anderson Cancer Center and Dr. Dusty Majumdar. As I said, we're going to cover liquid biopsies and the importance in the field of detection of cancer in early stages when it's most curable and where we stand today with the technology. Additionally, Dr. Hanash has very recently published a seminal article in the Journal of Clinical Oncology, where the largest blinded study of its kind reviewed a 4-protein marker panel that informs about the risk of harboring lung cancer and the need for CT screening. For those of you who are new here, my name is Don Davis, and I'm a consultant in life sciences focused on managing complexity and increasing the performance of organizations. So with that, let me introduce my guests. Dr. Hanash and Dusty. Dusty, did you want to kick us off and go ahead and start with a brief intro of yourself?
Yeah, sure. I would like to also mention we are honored to have Dr. Hanash join us in this conversation. Sam Hanash has been a pioneer and a leader in early detection of cancer, and we're really privileged to have him to opine on what's going on in this space. And Don, I've been a marketing and strategy executive now for over 2 decades in companies such as IBM, GE Healthcare. Had the honor of working in one of the early cancer detection firms, Exact Sciences as well. And I work with corporate America in different capacities at the moment, and also advise different health tech related startups.
All right. Thank you.
Well, let me introduce myself. So I'm Sam
please.
Hanash. I lead the McCombs Institute for Cancer Detection and Treatment at MD Anderson. And I've been in the field of cancer early detection for over 2 decades. That goes back to when I was at the University of Michigan. I was taking care of patients, and I had a patient who presented with advanced stage disease. I had to tell the family, unfortunately, there's nothing that we could do. The disease is advanced, and the family was very obviously upset by this and said, well, why did we wait until the disease is so advanced? Why couldn't we catch the disease earlier? And so that was my trigger to want to come up with the better mousetrap that we could catch cancer at an early stage. And that led to my working on wanting to develop blood tests for cancer early detection.
Well, I mean, it's exciting, and it's great to have you in this space for sure, Dr. Hanash. So to get us started in the topic, it seemed like quite often anymore, anytime I check the news for life sciences, or even, last week, I believe it was, there was a panel discussion at JP Morgan's event where they talked about liquid biopsy. To just get us started in the topic, can you talk to us a little bit about what liquid biopsy is?
Well, basically, the concept of liquid biopsy is that could you, with a biological fluid, which obviously is liquid, be informed about cancer? And so what could you be informed about cancer about? It could be basically somebody already has a cancer, and they're going to use the blood test to determine the molecular profile of that tumor, so that you could decide exactly how you should treat that tumor. There's been a sort of debate as to what would be better. Is it having a tube of blood that tells you about what's going on in the tumor, or a biopsy of the tumor itself? So there are advantages and disadvantages. So when you're dealing with a biopsy of the tumor to determine the molecular profile, you're looking at one tiny part of that big tumor, and you may not be looking at what's metastatic. You're looking only at the primary tumor, whereas the blood can give you sort of like a broader picture of what the molecular profile of the tumor is like. You could decide on the treatment, and then you could use the liquid biopsy to follow up and see like what sort of response you got and is there any residual disease. So this is kind of like from how you manage patients with liquid biopsy. The question then is also, could you possibly use liquid biopsy to catch cancer even before onset of symptoms? And so that's kind of the use of liquid biopsy for early detection.
And Dusty, do you have anything that you'd like to add to that?
Yeah, sure. I just want to kind of set the perspective here for folks who might be joining and perhaps not very familiar with this field, right? So it's been 5 decades since President Nixon declared the war on cancer, right? And the reality is that after 50 years, we are still not winning the war on cancer. Despite all the advances in therapeutics and screening, we're still finding cancer too late. Um, and we know that this year, 2022, nearly 2 million new cases of cancers are expected to be diagnosed, and some 610,000 people will likely die from cancer in 2022. And this is actually up from last year, which was 1.8 million new cases that occurred in 2021. So we also know because of COVID, screening's kind of taken a little bit of a back seat. For the first time, we see breast cancer death rates increasing among American women under age 40, for sure. And prostate cancer is being detected more and more today at a later stage than ever before. So, and we know the PSA test has largely failed us. So, this is the reason why we see tremendous interest in this field right now. We do have a lot of investor dollars flowing in. As we know, some of the clinical trials that are running right now that are heavily funded. And part of the unmet need is today we just have 4 cancers for which there are screening guidelines from USPSTF, the United States Preventive Services Task Force, at the population level. So that's breast, colorectal, cervical, and prostate. And if you combine all of these together, we're only catching 15% of the cancers early. So again, there's a huge unmet need here, and we're going to talk more about some of the different trials that are going on and what they are targeting. We're going to talk about the multi-cancer test as we go along this conversation. But I just want to set the stage, Don, at this point, and let you continue on this conversation.
Thank you
Well,
so
Dusty,
much, Dusty.
speaking of huge unmet need, I can list quite a few unmet needs. One is obviously better treatment. And so there's an unmet need for that. The other one is, like you talked about, an unmet need to catch cancer early. That's another unmet need. There's yet an even more important unmet need than catching cancer early, namely determining who is at risk of developing the cancer so that you could intervene even before the cancer is has developed. And how would you intervene? Altering lifestyle, or it could be that with all of the excitement about vaccines at the present time, if somebody is at a risk of breast, colon, lung, pancreas, ovarian, what have you, and you are able to determine that risk through a liquid biopsy or other means, then you can intervene. Here's the vaccine to prevent ovarian cancer that we think, based on your profile, you are at risk for. So, to win the war on cancer, we need to move across all of those fronts: better treatment, better early detection, better risk assessment, better intervention before the cancer through prevention. So, all of those things are going to be very important.
And Don, I would just add to that one more: better compliance. And Sam, knows this, right? For colorectal cancer, after all these years, the compliance is in the 65% range right now. It actually dropped a little bit last year. For lung cancer, with low dose CT screening, the compliance rate is around 15% after a lot of effort since the NLST trial that was held in the mid 2000s. So that's a huge unmet need. So the need for a simple, accurate test that patients can take at their convenience and they can believe in is critical. And that's something that's FDA approved and reimbursed. So we're going to come to all of those things as we go along, Don.
That brings up a good point, Dusty. The cost of cancer care and pharmaceutical drugs, does continue to grow every year. Do you believe that early detection will have a huge impact on reducing the economic burden of cancer?
Well, I do, and Sam is also in a great position to answer this, but I'll just be very brief, and
Well,
I'll let Sam expand
I
on
think
this.
early detection and catching cancer at an early stage has a tremendous impact on how you would treat the cancer. When you think about how we are developing drugs at the present time or therapeutics for cancer, you wait until somebody is advanced stage, has failed with so many other therapeutics available, and then you try to see if that drug is going to work. On the other hand, it may fail at that point during the progression of the cancer, and had you tried your test on early stage cancer, then it would have worked. And so we need to go back to the drawing board. If we're able to catch cancer in early stage, how would we treat that cancer differently? And what treatments that would have failed with advanced stage cancer might very well work at an early stage. So it would change the conundrum completely if we're able to catch cancer better at an early stage. But like I said, I'm more of an advocate of trying to prevent the cancer than having to worry about cancer at an early stage, just that part only.
Yeah, so I mean,
Yeah.
and certainly being able to know that you're at high risk to get cancer, similar to, cardiac disease or anything else, right?
Absolutely.
It'd be similar
Absolutely.
in my mind. So, sorry,
And
Dusty,
we
I
need
believe
And
I
to
Don,
interrupted
just
calculate
you.
to
risk for cancer, and we need to have the intervention at that point in time, whether it's vaccine or whether it's changing in the lifestyle or some other modality. And that's absolutely key.
Hey, Don, I'm getting a couple of pings saying that we may not be live at the moment. Can you just check, or it could be just an individual issue? Uh, Hopefully, we are live. Are you getting
Looks
any
to me,
comments?
it appears to me that we are live. I am not seeing any comments. No.
Okay. All right. So let's continue. I'm getting a couple of pings in that direction, but continuing on the theme that Sam just said, right? I was just looking at some numbers this morning. 5-year survival rate between early and late diagnosis is 56% versus 5% if you look across cancers. So, for example, when you look at renal carcinoma, um, when it's detected, when it's still localized in the kidney, 5-year survival rate is 93%, but the survival estimate decreases to 12% when the cancer has metastasized. Similarly, for 5-year survival rate for female breast cancer drops from 99% when detected locally to 27% when the cancer is spread. And I can go on for lung cancer and colorectal, but that kind of shows you there's a huge difference when you shift the detection to the early stages.
So I would like to recognize, though, too, that, something that I keep seeing in the news, over and over again is that, there are different articles that keep coming out, things like liquid biopsy being misplaced faith in early cancer detection. Um, you know, all this tells me is that people just don't understand really where we are today in terms of creating a liquid biopsy test, that does provide early detection. So I guess, I'm looking to try and get some comments from both of you with regards to the overall goal. Should it be, overall looking at detecting early stage cancer today, or should it be that we make some other improvement on getting the cancers that are already present in people? Dr. Hanash, why don't I turn to you first for that question?
Well, that's obviously a little bit of a complex question to answer in a sentence or two. The issue is uh, what is the purpose of a liquid biopsy for cancer early detection? And so, is it to actually detect the cancer or determine that somebody has a risk of having this or that cancer? It's a big difference. That's one issue. The second issue is we do have modalities available today for common cancers. We have CT for lung, we have mammography and other imaging modalities. And so the question becomes, and those are the common cancers, 5 common cancers for which we have screening modalities. So, in the face of those screening modalities, the question becomes, what is the purpose of a liquid biopsy, which in principle, if it's a multi-cancer, is supposed to detect those cancers when we have those modalities available? So that's a big issue. So is the liquid biopsy going to replace those modalities? The answer is no. It's an emphatic no. And so if liquid biopsy is not going to replace those, then what would it contribute for the common cancers? We'll talk about common cancers first, and we'll talk about the rare cancers next. And so it would seem to me that it is going to be very hard at the present time for a liquid biopsy to be competitive with the current screening modalities that have very high sensitivity at very early stages. And so the way I look at it is a liquid biopsy at the present time for common cancers would be best suited to determine the risk, personalize the risk. We know that there's a lot of issues now about screening for breast cancer. Then, do women need to be screened every year, every other year? What modality? If the mammogram is negative, should we have yet another mammogram every year? So, the liquid biopsy, as far as I'm concerned, would be the one to personalize that woman's risk for breast cancer. And so, if it looks like her risk is high, absolutely, you should get an imaging modality on a very regular basis. If it turns out that the risk is even below average, then maybe one could skip the imaging modality. That's an example for breast cancer. For lung cancer, as Dusty mentioned, uh, the compliance rate with screening by CT is very low. On the other hand, if there's a blood test that could tell you, well, look, your risk is very high, then you should get the CT, then compliance would be much better. So, in my view, a liquid biopsy for the common cancers should be one that empowers the current modalities available as opposed to competing with them. On the other hand, for the rare cancers for which there is no screening available, then that would be very beneficial, obviously, to have a multi rare cancer, pan cancer sort of liquid biopsy that would pick up, even if it were to pick up 10, 20% of those cancers for which there's no screening modality available today, that would be a plus. And so it's not like one size fits all. The liquid biopsy has to perform the same way across all cancers. It has to have multiple sort of performance criteria depending on the cancers that the multi-cancer or pan-cancer liquid biopsy is intended to detect.
Very good. Dusty, is there anything additional you'd like to add to that?
No, I think Sam covered most of it. I totally agree with him. In fact, this is something that Sam and I both espoused almost a decade ago when we talked about lung cancer screening as it stood at that time. And unfortunately, it has not really progressed a lot since then in terms of compliance. You still are in the 10 to 15% range for heavy smokers complying with this. So if there was a blood test that actually stratified the risk of these patients who are heavy smokers to go into lung cancer screening, that obviously would be a tremendous improvement on the current standard of care that exists today. Sam mentioned the rare cancers, right? And the challenge in the rare cancer space is the prevalence in some of these rare cancers is less than 0.1%. So somehow, if you could actually aggregate some of these rare cancers and you'd have a higher prevalence, so you'll have to screen less number of patients to find one of these rare cancers. And if there's a universal kind of a pan cancer test for these rare cancers, by aggregating the prevalence, you may be able to raise the positive predictive value even for a test where the sensitivity could be quite modest. So those are some of the things that we need to think about. And I totally agree with Sam that this is going to be personalized. It's going to be targeted in a way that we are actually getting the best outcomes for both the aggressive cancers that are fast moving, as well as some of the rare cancers and some of the indolent cancers. For example, with prostate cancer, men would probably die before the prostate cancer, in many cases, becomes metastatic. And we just have to keep in mind all these different nuances.
And I think you covered this early on, Dr. Hanash, as well in your opening comment. So how do you think the future blood tests are going to work for cancers? And, how do you think what about things where there is no screening protocol at the moment?
Well, I think the expectation of a liquid biopsy test for cancers for which there is screening available today versus cancers for which no screening, the expectations are radically different. And we cannot treat both the same way. And so how are they different? Now, if you're dealing with a rare cancer, then your ability to detect that cancer has to have tremendous specificity. Otherwise, because it's such a rare cancer, you're going to have a lot of false positives. And so, as a result of having very high specificity, that's going to reduce the sensitivity of your test. But one could argue, and this is an argument that Grail has made, for example, that those cancers for which there's no test available today, if we can pick up 5%, 10%, 20%, even that percentage is small, it's still better than zero. And so I buy into that argument at the present time. Hopefully, eventually, maybe tests would become available that have much higher sensitivity at a high specificity. But for other cancers, this should not be a requirement that you have 99% or 99.99% specificity. And I can give a reason for that. Right now, what does it take for somebody to be eligible for CT screening? It does not require that you have 100% risk of lung cancer. It requires that you have a maybe zero point 0.5 or 1% risk of lung cancer. That's enough to trigger a CT modality. So we need to look at the liquid biopsy for common cancers for which there is another screening modality very differently in terms of sensitivity and specificity.
And Dusty, anything additional you'd like to add?
Well, I think what Sam said resonates with me. Do we really need a specificity of 99.5% across the board for all cancers? The answer is probably no. If you work in conjunction with some of the screening methods which are already available, and at least in the short term, try to bolster those with a complementary liquid biopsy test. Um, I would also agree with Sam that for some of the other cancers, perhaps we could buy into the argument that we need a higher specificity. Um, and then the issue really becomes at such high specificities for stage one and stage two, are you really going to be able to detect anything with a reasonable sensitivity? And there's some mathematical arguments to that as well. Um, there are people who have shown in their work where the tumor is smaller than 6 millimeters or so, it's unlikely that one would detect by the techniques currently available circulating DNA, right? Because there probably would be in 10 mils of blood less than one genome present. And that goes up as the tumor goes to 10 millimeters and 15 millimeters, where you can, with reasonable certainty, detect some kind of DNA. So I think it's very nuanced, and if you're only depending on circulating tumor DNA, then your chances of success are even lower at the early stages of cancer. So when you start combining that with when you combine mutation and methylation in circulating tumor DNA with proteomics and fragmentomics, and perhaps even apply some kind of AI methodology to it, your probability could go up higher. It's good to see that this is going on right now. There are people who are seriously looking at this, and hopefully we'll see the advances coming very soon.
Well, I have to say that I don't buy into those theoretical calculations that if you're dealing with a tumor less than 1 centimeter or 6 millimeter or what have you, then it's unlikely that you're going to have one copy of DNA in the blood. I just do not buy into those. There's no evidence to that effect. It also totally ignores the fact that cancer doesn't happen overnight. It goes through progression from early stages, even before what we are now calling stage 1 cancer. It goes through an evolution from pre-malignancy to malignancy. And so at the pre-malignant stage, there could be a lot of changes at the DNA level, including mutations, what have you. And so if you have now, let's say through artificial intelligence, the ability to look at a wide spectrum of potential alterations in the blood that may not necessarily come out of that 0.5 centimeter tumor. They could be coming out of the 0.7 centimeter tumor, also coming about the pre-malignant tissue surrounding the tumor. And it could very well be that you're going to have an increment in mutations, and that would kind of empower whatever test you're using, whether it's a protein, or whether it's a DNA, or whether it's a mutation or methylation or what have you. So the argument that, okay, well, 0.5 centimeter tumor have only so many cells, and therefore you're going to have so many copies of DNA, I do not buy into that.
And if you go back to Sam's work in the past, there are studies where Sam has actually been able to show that even before radiologically you see anything in terms of a tumor, I believe it was a study in the lung, Sam, if I recall correctly, uh, they were seeing signals with proteomics, which clearly shows that there's a lot of things going on in the microenvironment of the tumor that you might be able to catch even before radiologically you can actually see anything.
Correct.
Yeah, I mean, that kind of brings up a good point. So, overall, there are a few tests with blood being used for testing reoccurrence and minimal residual disease. Why is it so hard to get early detection with liquid biopsy tests?
Well, I'm not sure by so hard if you're referring to the technology or referring to the implementation or referring to the cost effectiveness. There are many, many different factors that go into whether something works or not does not work from a kind of like public health point of view. From a technology point of view, it's certainly very hard. And because you're going to be looking, if you're dealing with a pan cancer test, at so many different variables in there, and it's like catching a tiger by the tail type of thing. And so for the longest time that we have been working on wanting to develop liquid biopsy tests for cancer, our sort of kind of vision was a lot more modest than a multi-cancer test. Let's first demonstrate that we can work for one cancer in an effective way, and then we can see, well, what about 2 cancers, 3 cancers, what have you. And so it certainly created a lot of shock when this notion came out that we can have a test that could look at X number of cancers at the same time. For us, kind of in the academic, scientific, low budget sort of mindset, then let's kind of move one step at a time as opposed to kind of take a big jump into pan cancer. But I have to commend whoever is working on pan cancer that they were able to attract the funding, attract the interest and the resources to be able to do that. The other thing is when people are bashing like the pan cancer, they are expecting perfection. And the way I look at it, it's kind of like basically the spirit of St. Louis crossing the Atlantic for the first time. I mean, don't expect a jumbo jet to be crossing the Atlantic the first time. I think one has to look at it from the point of view of the promise, and you're going to have, let's say, an evolution of those tests progressively. So I think it's commendable that there's interest in pan cancer. That's kind of like how I look at it, as opposed to wanting to bash it.
Yeah. Yeah, and I mean, I certainly can see that as well. I mean, and that's where I definitely give credit to a lot of people in this space, right? Is that, it's not like we're waiting until everything's perfect before we get to that, end point of having a pan cancer test. There most likely is going to be sort of an evolution to your point, Dr. Hanash.
Yeah,
So
well, I was involved in the Cancer Seek and made a contribution to the proteomics part of Cancer Seek. And when the paper came out in Science, then I was flooded with emails and calls from people, statisticians and others. What do you think that you really can detect cancer, pan cancer this way? This is not going to work. I told them, well, look, this is the first step. This is a proof of principle that maybe adding protein to DNA can have a significant contribution to cancer early detection. Not that we are ready today to launch a test of that sort.
yeah. And I think that's what a lot of the news articles that I'm seeing, at least, that they're at least indicating that, look, it's not going to be tomorrow. We don't know how far out it is, but it's not going to be tomorrow that we have this test.
Well, I wouldn't say it's not going to be tomorrow. I would say the first kind of Model T that's going to come out is not going to be the same as what you're going to have a few years down the road. That's kind of like how I look at it. We may end up with Model Ts at the start, but that's still a good start to have a Model T.
Right. So how would a liquid biopsy test be integrated into the clinical pathway?
Um, that's a very good question at the present time. The question is going to be how much buy-in one would have for a pan-cancer liquid biopsy test. It remains to be determined. There
Okay.
are believers and not so believers at the present time, as we all know. I've been impressed in the past as a physician scientist as to what it takes for something to be adopted. I think uh, marketing, publicity, shock and awe have a big impact on whether something gets adopted or not. We have, for example, known about hemoglobin A1C from a science point of view for a long time, and nobody was using hemoglobin A1C for monitoring diabetes until somebody decided to kind of publicize it, market it, and go out for it, and then all of a sudden it became a standard. And so, what it takes for something to be adopted requires a kind of sort of a way to get people to buy into it.
I would add to that two things. One is understanding the clinical workflow right now for at least the cancers which we have screening for, the four cancers that we mentioned previously. So how do you integrate a blood test, for example, into the clinical workflow for low dose CT for high risk lung cancer patients, right? I mean, or people who we suspect may have lung cancer. I think that is something that's very important, integrating in the workflow. Similarly, colorectal cancer, the compliance rate is around 65% right now. So we have Cologuard, the stool test. There are blood tests that are probably coming out. How do you again integrate that into an annual physical, or do at home test and do it effectively and compliantly? The other thing that is going to become more and more important as these multi-cancer tests come up is that today we don't have any legislation that actually is going to provide coverage for pan-cancer or multi-cancer screening. There's a bill in the Congress right now. It has bipartisan support for multi-cancer diagnostics and innovation in cancer diagnostic development, and that is going to go through the Senate and the House hopefully in the next few years, uh, or may not. So there is still some uncertainty with that. So I think the government angle of this is still up in the air, whether it's going to be a multi-cancer test is going to be truly covered or not.
Yeah, and it looks like we've gotten a couple of comments, back on the YouTube stream now. So I do know for sure that people are watching there as well. Dr. Hanash and Dusty. One of the latest comments that came through, even during your comment, Dusty, was that we'll start to see this whenever insurances start to pay as well, for the test
Mhm.
in the clinical pathway.
Yeah, and just that everyone understands, this is not an easy process. So some of the companies that Sam and I mentioned, Grail and Freenome and Guardant, are in the middle of clinical trials. These typically would take 3 to 5 years, depending on what kind of endpoint you decide on. After that, it could be a year till you get FDA approval, and then there's a CMS reimbursement that comes sometimes along with the FDA approval if you go through a special path. Otherwise, it could take another year for that. Once you have CMS reimbursement, Medicare would pay for it, but then the private insurance depends on the ratings that you get from USPSTF, United States Preventive Services Task Force, which only meets every 5 years. So if you got to really be at the right cycle at the time that the USPSTF is going to be meeting to show all your results and get their blessings for an A or B rating for private insurance to start paying for screening tests. So we're looking at a long journey here, and we can't just sit on our hands between now and then and not have some of these single organ tests moving forward in terms of integrating with the clinical workflow, improving the performance, and improving the compliance.
So one argument that's been made for cancers for which there is no screening today is that any test that sends people to other tests would be helpful. What kind of performance do you feel the FDA will demand for these types of cancers? Also, are there downsides that you believe that are here? And, in my mind as well, false positives that could flood the healthcare system might be another concern, for these tests. Dr. Hanash?
Well, yeah, for any test, uh, I would imagine what the FDA would consider is what's the consequence of that test being positive? What's the consequence of that test being negative? Now, if a test has a lot of false positivity, but if you are positive, you're going to go through surgery, that is a substantial, obviously, concern. Now, on the other hand, if your test could have false positivity, but as a result of that test, you're going to have some kind of imaging modality, then that's not as a severe a consequence as having to go and get an exploratory surgery of some sort. So I think those are things that I'm sure the FDA would take into account: the consequences of false positive and the consequences of the false negative. The FDA is not going to care about how much the test costs or what have you, but those are the two most important issues to deal with.
Dusty, anything else to add there? If not, I've got an AI question for you that I know is right up your alley.
Well, I would agree completely with Sam. And again, looking at some of the results that are coming out in various publications, especially for early stage cancer, stages 1 and 2, where for very high specificities, we are seeing sensitivities across the board of around 20 to 30%. I think the thing to worry about there is the positive predictive value for some of these individual cancers. Now, the advantage of aggregating some of these rare cancers and cancers with low prevalence is that you get an overall blended number of high prevalence, which is going to improve the positive predictive value. So I think that's the advantage of a multi-cancer test. And the other way to improve the positive predictive value or lower the chances of false positives is to target the right populations. Right? We know cancer is a disease of older people. So, you test people over 50, over 60. We know that patients, who have a family history of certain types of cancer are at a high risk of cancer. We know smokers are at a high risk of cancer, not just lung cancer, but across the board, bladder cancer, in some cases, pancreatic cancer. So, in some cases, we have seen that organ transplant recipients are at a higher risk of cancer. So targeting those populations with the right kind of tests, with the right performance, would be the key.
Yeah, absolutely. I'm going to stay with you just for a second, Dusty, because this next question I think is something that you could start with, and then maybe come back to Dr. Hanash on. So we hear some companies are now starting to talk about more and more using artificial intelligence to integrate part of the effort for liquid biopsies in detecting early cancer. What are your views on this initiative?
Increasingly, I think you see companies talking about that, and I think it's inevitable as you move into the multiomics arena that you may need AI to help you really get to the answers quicker in a more efficient way. Um, uh, Grail talks about using AI in their gallery test. From what I've heard, Freenome has been promoting AI in their testing quite aggressively. But one of the discussions that Sam and I have had on this is okay, so even if you have AI helping you to predict or to detect cancer early with higher specificity, how are you going to create an assay based on an AI algorithm? And I'll let Sam talk about this because he has put some thoughts into it. Sam, go ahead.
Yeah, I mean, when we are talking about AI for any application, one has to distinguish the use of AI for discovery versus the use of AI for implementing the test. And so, if you're looking, for example, at DNA methylation and you have millions of sites of DNA methylation, and you have so many variables, the age of the patient, or the subject, or what they had the day before, or what medication they are on, it becomes a very complex undertaking to want to determine what would be the best indicator for this particular cancer. But once you have come up with the signature, then the AI goes out the window. You already have one, two, three, five, 10 particular features associated with that cancer. So let's not mix AI for discovery versus use AI to determine if that person is positive or negative. You're not going to use AI to determine if they are positive or negative. You're going to use AI to determine the signature that would make that person positive or negative. But once you have that signature, AI goes out the window.
And so what would the fallback be then? So what would happen? What would happen after you're finished using AI? How would that work?
Well, let's say that for a person with certain characteristics, for example, age, sex, and what have you, then this particular signature that we found, if it's related to methylation, meaning those CPG sites that are methylated indicate the presence of this cancer, then basically you have come up with that signature that you would implement when you are using the test to screen for this cancer or multi-cancer type of thing. And so it's very valuable. I can give you the example of back maybe more than 10 years ago. One of there are so many different things in the blood that can tell you about cancer. And a laundry list, you could look at DNA, multiple features of the DNA. You could look at RNA, you could look at protein, you could look at metabolites, you could look at the immune response. And back more than 10 years ago, I led a project that was supposed to be looking at DNA methylation. And it became so complex that I would think without AI, you would not be able to come up with the signature. So we gave up on this. And to the credit of Grail, that basically they went full blown approach to look at all those millions of sites and be able to come up with signature that not only tell you about the presence of cancer, but where the tissue is. And so I'm very envious of what they've been able to do at the DNA methylation, which we could not do without obviously not only the AI, but all the massive resources that went into that particular endeavor.
Very good. And Dr. Hanash, I'm going to stay with you for a second. So, in your most recent paper on a blood-based biomarker panel for risk assessment for lung cancer, it demonstrated how to identify individuals with a high risk of lung cancer. Can you tell us more about your key findings?
Yeah, the premise of our study, which that paper is the last in a series of papers that we have published, and like I said, we are the academic. We move very slowly, incrementally. If you finish one study, you learn from it, you go to the next, and you go to the next. But the premise has been that as of today, not even 10% of subjects destined to die of lung cancer are being basically are going through screening for lung cancer. And the question is, can we come up with a blood test that would determine somebody's risk of having lung cancer, or basically over a period of time would be diagnosed with lung cancer? And so that's what we try to accomplish in that study, namely going through the multitude, like I mentioned, the laundry list of different markers. We looked at all of them, just about, and then at the end came up with a simple test, 4 protein marker panel, that would determine somebody's risk of having lung cancer. So the concept is that if you are a smoker, for now the test is only applicable, the data that we have is only related to smokers. So if you are a smoker and you go to see your doctor, and the division is that, okay, we have a test that could determine your risk of having lung cancer. And if the risk is high, then through shared decision making, then you can decide whether you want to go through screening or not. And the major reason is that compliance with lung cancer screening at the present time is very, very low, number one. And number two, there are smokers who don't meet the criteria that may be destined to develop lung cancer. So if you have a blood test that could determine your risk, it may sort of persuade you to go through screening with CT. So the implementation of screening and the compliance could go up considerably, and we would save more lives that way.
And so, I mean, I'm taking then from that the overall premise being that if you did the protein panel, you have a high indication that you just need to be screened for lung cancer, then you believe that people will go for the lung cancer or for the CT
Yeah.
screening?
And
Yep.
so
Yep.
if you are eligible but undecided, having your personalized risk as opposed to some kind of a yes or no risk based on the criteria today, a personalized risk would be more persuasive for wanting to get the CT screening, number one. Number two, if you are less than a 20 pack year smoker, you're not eligible for screening. And yet we know quite a few lung cancer cases are among subjects who are not 20 pack year smokers. So if the blood test can say, even though you have smoked only 10 pack years, you are just as much as a risk of lung cancer as a 30 pack year smoker, then you should get the CT screening.
Yeah, it's a call out for all of my followers that are in this sort of group of people that carry around those white ribbons, right? I know that kind of the one guy that started all of that more or less wound up with lung cancer without being a smoker, and so he just said, look, I wound up with this. It wasn't something I felt like I could publicly even talk about. So if there was a way to screen people and at least say, to people like him that, hey, you're a high prevalence of needing at least to be screened, additionally, just to make sure that you don't have lung cancer would be wise.
Well, we also have to keep in mind that in most countries, there is no adoption of low-dose CT screening for lung cancer. On the other hand, if a blood test says you are positive, then that could be a clinical indication to get the CT screening.
Yeah, absolutely. And do you see a greater number of academic industry partnerships coming in this space, for liquid biopsy?
Yeah, absolutely. I mean, there's a lot that we bring into it. Like I said, I'm not the only one who's been in this field for a long time. I've gained a lot of experience, and so we have been preaching about all the different applications, the nuances of what one would do, and at times I feel that one size fits all in terms of what you need in a multi-cancer panel for lung versus colon versus breast versus a rare tumor. You cannot treat them all the same way in terms of having a requirement for the same sensitivity, the same specificity. So there's a lot of experience that we have had that would help in terms of how to sort of finesse a pan cancer screening test.
Very good. Dusty, do you have anything additional you'd like to add to that?
No, I think we covered most of it. I'll just add one more thing. So, why do we have all these companies that are jumping into it, and why are investors pouring in so much money into it, right? So, it obviously has to do with the market size that people see in some of these early cancer detection opportunities. So, for example, in colorectal cancer, if you take patients or if you take individuals between 45 to 84 years of age, there is a $20 billion market just in colorectal cancer. In lung cancer, if you and as you know, in colorectal cancer now, there are trials going on in blood from Guardant and Freenome. I believe Exact Sciences is also developing the next generation Cologuard, as well as looking at blood. You do have similar kind of situation with lung cancer. If you just take the high risk population, people who have smoked over 20 or 30 pack years, so that's smoked at least a pack for 30 years or two packs for 15 years, that's 30 pack years. You can easily see a market of 30 to 35 billion. And this doesn't even include folks who are the non smoking population and women who have never been smokers, non smokers, are increasingly getting lung cancer now. So you could also imagine a test coming to that arena. Um, you also see in pan cancer, if you look at what's going on across the board, if you look at all these different cancers, you could easily see a market opportunity of around 50 to 80 billion dollars, depending on whose calculation you really believe. So there's obviously a very attractive space across the board, and that's why you see a lot of the major players jumping in, like Grail and Illumina and Guardant and Exact Sciences and Freenome, and there are probably a list of around 20 other players that we are not mentioning here.
Yeah. Very good. So one of my favorite things to talk about as well is what's next. So, Dusty, why don't we start with you since the camera's on you? Where do you think we'll see the next advances come in liquid biopsy?
Well, I think that really depends on how the clinical trials, which are underway, pan out, right? I think that is the key. And you know, if you want, I can talk a little bit about what we are seeing in that space. But I think we are seeing all the right things happening in terms of the combination of mutations and methylation and proteomics coming together in some of these tests that are being developed or at least being talked about. And then the advent of AI, may be able to help in some way to deal with the complexity of data. And Grail has been pretty public about leveraging AI, and so has Freenome, and I'm sure the other players are working behind the scenes to leverage AI in a way to really figure out the way you can detect cancer, especially at the early stages when it is most treatable and curable. So I see that continually happening. And I sincerely hope that we don't wait for a pan cancer or a universal cancer test to become perfect, but continue the journey that we have with some of the tests that are already available and complement them with a blood test so that the overall compliance goes up, the accuracy of screening goes up, and we reduce the mortality of some of these very lethal cancers that are still taking a lot of lives every year.
Very good. And Dr. Hanash, what do you think's coming
Yeah,
next?
well, basically, the way I look at it is there's going to be one or two or three or four pan cancer tests out there. And I can guarantee you, if Company X is working on their first pan cancer test, as soon as they put it out, they're already working on version 2.1 of that cancer test and 3.1 down the road or what have you. So there's going to be an evolution of tests out there with uh, improved performance over time.
Very good. And essentially, that's what I have for today. I'm glad that both of you joined me for this liquid biopsy panel discussion. So thank you very much for your time. And as always, I will essentially release the audio version of this on an audio recording as well, so that people can watch this if they'd like to on YouTube or LinkedIn, but also go to their favorite podcast platform to understand more. If you want to connect with any of the guests here, you could see them on my website at www.lifesciencesuccess.com. Thank you so much for watching. Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.